Polymerase chain reaction, otherwise known as PCR, is the process of amplifying and making multiple copies of DNA for research or production purposes. So as mentioned, gel electrophoresis is a way of visualizing our DNA and matching up the criminal to the crime scene, if you will, or figuring out which genetic similarities or differences occur within two different populations or two different people. But if we don't have very much genetic material available, we can undergo PCR to help duplicate or make multiple copies of DNA. The components of PCR include the DNA sample, some primers, nucleotides, so that again comes from our DNA, Taq polymerase, which is a very rare particle that we use in DNA in PCR, a mixed buffer solution that helps kind of break up the different DNA components, and then obviously a tube to keep everything in for our PCR. And then we put this DNA sample and this PCR mix into a thermal cycler, and the thermal cycler helps makes the DNA undergo a very specific cycle. There's a couple of cycles that they undergo where it heats up the DNA really, really hot and then cools it down, and it goes back and forth throughout. And it kind of goes on and on for a lengthy period of time. And this helps us to make copies of our DNA. This video is going to explain it a lot more eloquently than I just did. So take a look.
A polymerase chain reaction uses heating and cooling cycles to amplify small samples of DNA to workable quantities for further study. A double-stranded sample of DNA is obtained and then denatured, or split into two strands, at 94 degrees centigrade. The denatured DNA is then cooled to 55 degrees centigrade, and selected complementary DNA primers are annealed, or joined, to the region of now single-stranded DNA that is desired to be amplified. At 72 degrees centigrade, the special enzyme Taq polymerase extends the single-stranded DNA by complementary base pairing of deoxynucleotides in the form of the 4 DNA bases. This represents one cycle of the polymerase chain reaction. The cycle is repeated 20 to 30 times to obtain millions of copies of the original DNA sample. For example, 25 cycles will yield 2 to the 25th power, or 33,554,432 copies.
So again, in PCR, we undergo many different copies of the deoxyribonucleic acid, or the DNA, to help make us have multiple copies available for genetic research purposes. So that is the two laboratory techniques that we're going to focus on today. Again, that's gel electrophoresis in the first video and PCR here in the second video. Tune in for the next clip to learn a little bit more about reproductive technology.